Welcome back, everyone. Today's guest is a man who hardly needs an introduction, but I'll give it anyways. It's Dr. Aubrey de Grey, who's one of the first people to come to mind when people think about longevity science and lifespan extension. Dr. de Grey has been at the vanguard of the fight against aging for decades. He has co-founded multiple nonprofit organizations, like the SENS organization, and more recently LEVF, the Longevity Escape Velocity Foundation, and he's written multiple books on the topic, including Ending Aging. He's a prolific speaker who's presented at many international conferences and events. I was truly honored to have him on as one of my first interviews. This, by the way, was recorded on January 20th. Please welcome Dr. Aubrey de Grey. Okay, thank you for coming on. I appreciate it.
My pleasure.
My first question is, in 2021, you tweeted, "I now think there is a 50% chance that we will reach longevity escape velocity by 2036. After that point, the methuselarity, those who regularly receive the latest rejuvenation therapies will never suffer from age-related ill health at any age. Can you explain to the listeners what makes you so optimistic, particularly when this is a problem that we've been trying to address since essentially the dawn of civilization?"
Well, I guess you want me to do that in a concise way, which is not trivial. So, essentially, whenever any big, dramatic technological breakthrough occurs, it is preceded by a lot of stuff that doesn't have any real, visibility. Something like powered flight, for example. Nobody could know, no random person in the street could know that was going to happen. But the people who were working on aerodynamics and, things like lift and so on, they were putting the bits together, and they kind of knew that there were, ways forward that might work. So it's the same in this situation. It's really only the people at the cutting edge, the experts who are looking at all of the various necessary component technologies and advances that would be required. It's only people like that can really make an assessment of this. Now, of course, even then, the assessment is very speculative, because you never know what kind of surprises you're going to encounter in the run-up to actually bringing the thing off. So that's why I always give my predictions the way you've described them as probability. I think there's at least a 10% chance that we won't get to longevity escape velocity for 100 years. That doesn't bother me, of course, because a 50% chance is quite enough to be worth fighting for. But yeah, it's really there's no simple answer to your question. It's just I'm at the cutting edge of all of these details, and it's all coming together.
Mm-hmm. I believe it. I ask more for the people who might be skeptical. And I'm curious to know, since this is something that you said about 4 years ago now, has your estimate shifted? And if so, what would you place it at now?
It hasn't really shifted, no. I think, maybe 2036 to 2038, that kind of period. Obviously one can't be precise about this, but yeah, that kind of estimate is still about there. We've had a few bumps in the road in terms of funding, which might have slowed things down by a year. And the people who are responsible for that know who they are. But other than that, not really.
Mm-hmm. I imagine that AI advances have helped things along quite significantly, or maybe not yet.
Certainly AI has been helping already and will help much more in the future, and that kind of factors into how I get to my estimate. The big advances that we've seen in AI, especially the ones that are, benefiting the whole of biomedical research, not just aging. So, like, for example, AlphaFold, that's the most conspicuous one. These things are going to continue to happen. We're now on the cusp of having programs that can design proteins from scratch. And indeed, we're not really even on the cusp of it. It's already happening. Of course, the thing about what we can extract from nature is that everything that exists in any living organism got that way by evolution, which works in small steps. And in silico, once we have the understanding of how to build a protein of a given shape, we don't need to worry about that. We can build something that's got a lot of differences from anything that's ever evolved. Just last week, there was a big announcement. It came out in MIT Technology Review of OpenAI and their collaborating company, Retro Biosciences, figuring out how to make versions of what are called the Yamanaka factors that are much more potent than the natural one. This is the kind of thing we're going to see more and more and more.
Mm-hmm. Now, the framework that you've embraced and revolutionized, I think, for how aging works is that people are like cars, basically. They break down and sustain damage over time, and you just have to keep fixing and replacing the parts. You've outlined 7 types of damage in SENS. If you had to allocate 100% of global aging research funding across these categories right now, what would the distribution look like and why?
That's a very difficult question to answer, mainly because the distribution changes over time. Every time a piece of progress is made, you have a new experiment to do, a new piece of progress to make, and typically the later ones cost more than the earlier ones. However, so against that, you have the issue that every step forward is a de-risking step, which means that sometimes it's easier to get the next aliquot of money, even though it's more money, just because people are more willing to write the check to take that risk. So it's very case by case. At the moment, the main focus that I have is actually not as between the various different components of the damage repair approach, but rather the thing that we're doing at LEV Foundation, combining things that already somewhat work in mice and making them jointly work better. That's something that nobody else is doing, not in earnest anyway, not with cell therapies and gene therapies the way we're doing it. And, the sooner that changes, the better. But it's extremely hard to get money for. It doesn't really create very strong new intellectual property. It doesn't really test hypotheses, so it's hard to get money out of academia for it. And the experiments are pretty expensive, like the one we're just coming to the end of now involved 1,000 mice, which is a lot of money.
Yeah, it's very long term, so it doesn't exactly work with the incentives in academia or in industry.
Right.
I was surprised to see that you don't list telomere shortening as one of the primary types of damage. Is this because you see it as more of a protective mechanism than as a cause of aging? And what is the evidence you've seen for this that informs your position on that?
So, telomere shortening is something that is often oversimplified when people talk about it, especially as it happens in humans. Firstly, we have to remember that nearly all of our cells in the body are either constitutively incapable of cell division, because, in other words, what's called post-mitotic, like neurons, for example, or heart muscle cells, or else they can divide, but they actually only do divide very, very rarely when there's an injury or something like that. So these are cells whose telomeres do not appreciably shorten during life, and as such, would not benefit from re-lengthening of those telomeres. If we look at the exception, then they are the stem cells of rapidly renewing tissues, or regularly renewing anyway, like the blood or the lining of the gut or the epidermis, the outer layer of the skin. These are cells where we do see shortening of telomeres during life. However, it's very, very, very slow. And the reason it's slow is because the stem cells that perpetually replenish those tissues, those cell types, are actually not subject to what's often called the Hayflick limit. They make a very small amount of this enzyme telomerase, which naturally extends telomeres. So they basically put off the shortening of telomeres by a certain amount. Now, you may ask, well, why don't they do it a little bit more so that we don't get any telomere shortening? And you have indeed already answered that question in your question. It is believed, anyway, widely to be protective against cancer. The less telomerase you make, the more the cell that's trying to be a cancer has to upregulate that expression of telomerase in order to maintain its telomeres during rapid cell division.
Mm-hmm. Do we need some way to maintain telomere length in healthy cells while preventing cancer cells from doing the same?
Yeah,
how do you
probably.
maintain that balance?
Probably. Yeah. So this is absolutely the right way that I look at it. That it's a balance, as you say, and we have to find some way to get the best of both worlds. Essentially, we have to do one thing to cancer cells and another thing to non-cancer cells, but that's really, really hard because cancer cells are very good at, if you like, masquerading as non-cancer cells. The best news in that regard right now that I know of is a fantastic technology being pursued by a company in Chicago called Maya, M-A-I-A. Comes out of a professor out of a lab in Dallas, Texas. And essentially what it does is, well, in shorthand for people who know the terminology, it turns telomerase into a suicide gene. So you're going to have listeners who don't know what a suicide gene is, so let me spell that out in a little bit more detail. When telomerase is expressed, when you're making this telomerase, it extends the telomeres, it adds new sequence on the end. And, of course, that sequence consists of nucleotides, A, C, G, T, same as every other part of the DNA. Now, the drug that this group in Texas discovered is a drug that is rather similar to one of those bases, thymine. And the only difference is it's got a sulfur atom in it. Sorry, guanine. I got that wrong. So 6-thiodeoxyguanine it's called. So it's got a sulfur atom in it. And that means that if it's incorporated into the DNA is messed up. It's recognized as damage. The cell can't repair the damage. The cell dies, basically. Now, it turns out that this particular analog of guanosine does not get incorporated into the DNA during regular DNA replication or during DNA damage repair, which involves replication. Only telomerase does it. Telomerase kind of doesn't see the difference, doesn't spot the sulfur atom. And so it incorporates this thing into the telomere. And that messes the telomere up and the cell dies pretty quickly after that. So cells that make a lot of telomerase, you give them a bit of this drug, they are screwed. As I mentioned, there are cells in the body that make telomerase, but they make very, very little of it. So you've got a nice therapeutic window, as it's called, in terms of the dose and the duration, where you can give someone who has cancer, even late-stage cancer, this drug, and its cancer will fall apart and die, and the stem cells won't be affected, because you don't need to do it for too long. If you combine it with immunotherapy, which is what Maya are doing, then it's even better. So I have high hopes for this kind of thing. And if you can do that, if you've basically got cancer completely under control as a result, then you can afford to upregulate telomerase, whether pharmacologically or genetically, in other cells. Not at the same time, of course. Once you've got rid of the cancer, you do it then, and so that the drug doesn't do any harm.
I see. So you believe more in damage repair. That's the aspect or the framework that you focus on more. And I think you've been somewhat skeptical of epigenetic reprogramming approaches.
Not exactly. No, let me stop you there. I think you may have slightly misunderstood that. Epigenetic reprogramming is very, very interesting. The partial reprogramming that is so fashionable right now in vivo, where you give these Yamanaka factors to mice or eventually humans. The only thing that's difficult about it is the cancer problem, because essentially when you're doing this in cell culture or even in a short-lived animal like a mouse, you may not see cancer, but you may be inducing the early stages of cancer, and for a long-lived organism like you and me, that really matters, and nobody can tell at this point. The difficulty is that in the body, in anyone's body, yours and mine, there are lots of cells that are most of the way to being cancerous already. They've got most of the necessary mutations. So, in other words, the partial reprogramming factors may kind of push them over the edge into being properly cancerous. But this is one of the things I was referring to when I was talking about upregulating telomerase once you've got cancer really under control.
What's your most controversial technical view about aging that other respected researchers disagree with you on?
Well, that used to be an easy question to answer because, of course, the whole damage repair paradigm was a real paradigm shift. When I first started talking about it back in 2000, 2001, hardly anybody understood anything that I was saying. And to the extent that they looked at my conclusions, which were, of course, very radical in terms of the potential in terms of life extension, they thought, this guy's gone crazy. They already knew that I was smart. I'd been around in the field for a few years. So the view was kind of, he'll come to his senses in due course. But, of course, I persisted. And eventually people got the message. And, once the Hallmarks paper came out, which was 2013, about a decade after I started publishing this stuff, it all became mainstream. So now my views are not really considered particularly controversial as regards to the technologies that might be required, the damage repair approach, the divide and conquer nature of it. What is still controversial is the whole concept of longevity escape velocity. But that's honestly because, it's a concept that makes predictions about longevity, and most of my colleagues are too politically correct to want to think about that, so they run away very fast when one talks about it. But they don't have any actual arguments against what I say.
There's ongoing debate, it seems, about whether removing senescent cells could deplete stem cell pools or disrupt tissue maintenance. Are there specific molecular markers that would let us better distinguish between beneficial versus harmful senescent cells? And if so, how close are we to that precision?
Yeah, great question. Well, so it's very much a research area still, so I would definitely not say there are reliable such markers. In fact, it's actually not trivial even to decide whether a cell is senescent at all or not, because the markers we have for that are not 100% universal. However, what we can say is that the body tries its best to get rid of senescent cells already. We know that the immune system identifies and removes senescent cells, kills them off, and the only reason they accumulate during aging is because the immune system is not 100% effective at doing that. So, what this means is that it's probably not the case that the immune system somehow goes after some senescent cells that are bad and leaves the senescent cells that are good alone. It's probably the case that all senescent cells are bad if they hang around too long. And that what ought to happen is, insofar as senescence is an adaptive phenotype that helps with things like wound healing, for example, once the wound has been healed, the senescent cells ought to all go away, but they don't all go away. So this means that, yes, senescence is a feature, not a bug, so to speak, but if we just were to augment what the body already does, either by making the immune system better at removing senescent cells or by using other ways to remove them to augment what the body does, then we won't be doing any harm. We just want to not do it when you've got a wound, for example.
I see. How does gene therapy fit into the SENS framework? And for those who don't know, maybe it's worth briefly explaining what gene therapy is.
So gene therapy comes in various flavors, but essentially the idea is usually that you insert a new gene into the genome of cells in the body that will make a protein or whatever and restore or enhance function of some kind. This gene may be one that most of us have naturally and that is congenitally mutated in the people you might give the gene therapy to, for example. That's the main use of gene therapy these days. But potentially it can be used to introduce genes that are not human at all, genes from other organisms, for example, that may be able to perform functions that we just don't have. That's an approach that I've pursued in relation to the number one cause of blindness in the elderly, macular degeneration. And that went rather well. We pursued it in my foundation for a long time. Eventually we made enough progress that we could spin it out as a startup company. That way it got rather more investment, and they carried on, and now it's about to be sold to Big Pharma. It's a genuine, it's a thing. There are other types of gene therapy now, though. In particular, there's this thing called gene editing, which is where you use various techniques to modify the existing sequence, maybe just change one base pair, something like that. And this is usually done these days using a technology called CRISPR, which is essentially leveraging the adaptive immune system of bacteria. That's how it's normally described. CRISPR is a complicated, and it was initially a somewhat high-risk approach. Essentially, it would do things you didn't want it to do quite often, even if it was also doing the thing you wanted it to do. But people have, of course, worked on that over the past decade and have made it a lot better. So it's now a very versatile technique, and it's used for other things in addition to gene editing. So this kind of comes out under gene therapy as well. All right, so your question was, what is the relevance of all of this to the damage repair approach? It's huge. I've just mentioned one example, the example of macular degeneration. But in general, if we have a function that we'd like to introduce into the body, this is how we would do it. And if we want to, for example, alter the sequence of a gene that seems to be working well, but we want it to work better, or we want to make it work in a different way, then we may be able to find out how to do that in cell culture, and then we want to implement that by gene editing in vivo, and CRISPR is your friend in that regard.
Tell us about the RMR study, the flagship study of LEVF that's in progress right now.
Sure. So the damage repair approach is inherently and inescapably a divide and conquer approach. The body is a ridiculously complicated machine, and therefore it damages itself like any machine does in the course of its normal operation, but it damages itself in a huge number of different ways. Now, the body has evolved ways to automatically repair some of that damage. But the way that evolution works is such that types of damage which accumulate really, really slowly and are only bad for you when you're old are ones that evolution doesn't bother to repair as they happen, because by then evolution has passed on our genes through reproduction. Okay, so we've got all these types of damage that are accumulating. When I first started talking about damage repair, I classified those types of damage into 7 major categories, which is a useful way to understand how we might develop the damage repair approaches, because within each category there are lots of things in common with the damage repair. Like, for example, stem cell therapies or partial reprogramming can potentially address all of the loss of stem cells that we see, even though we see that loss in many different organs. And, of course, it's different cell types in different organs. But anyway, the point is that even though there's this classification, it's still very much a divide and conquer approach. So any divide and conquer approach in any technology has 2 phases. You've got to develop the components, and then you've got to put them together. Now, the putting together part in most areas of engineering is the easy part. If the individual components are all doing what you want them to do, then you can be pretty sure that just putting them together as you had originally conceived is going to work. In the case of medicine, though, it's not like that, because the body not only is ridiculously complicated, it's also, we don't understand it very well. In fact, we understand it extremely poorly. So that means there's quite a chance of getting surprises when you put the bits together. Now, the question then is, how do we start? Well, obviously we start in mice, same as we do with everything else. So over the past 20 years or more, I've been pursuing research in developing the components. Especially I've been focusing on the most difficult components, because those are the ones that were being neglected by academia and industry. But now, since that's all gone quite well, and a lot of these things have been spun out into the private sector and so on, it's time to start looking at putting these things together. And the ones that can be put together at this point are the ones I was not working on, the lower hanging fruit that were relatively easy and that other people were developing. But there's enough of those things now that are working individually to a certain extent in mice as damage repair. So you could take middle-aged mice, you give them this treatment, they live a bit longer than they otherwise would. There's enough of those now that it's time to start putting them together. And so 2 years ago, we started the first study of that nature that had ever been done. Involved 1,000 mice, cost $3.5 million, And we put 4 things together. One of them was not a damage repair approach, actually. It was just the kind of gold standard non-damage repair approach. It involved something called rapamycin, which is what's called a calorie restriction mimetic. But the other 3 things were indeed damage repair. One of them, coming back to one of your early questions, was telomerase gene therapy, to upregulate telomerase. One of them was to rejuvenate the blood stem cells by giving these mice stem cells from young mice. And the third one was a senolytic, a drug that kills senescent cells. And the study is nearly complete. There are only, as of today, there are 3 mice left, so I'm told. And it's gone pretty well. We haven't, knocked it out of the park. We haven't got, doubling the mouse lifespan or anything. But we have, definitely shown additivity, even with only 3 damage repair interventions. So we're desperate to do another experiment, and more than one, lots more experiments of this nature, but more ambitiously. I want to put 7 or 8 things together in the same mice at the same time, and I believe that we can make very big breakthroughs that way.
Well, I'm glad that's going well. Small side question, you mentioned how evolution doesn't really care whether we live longer because we just need to live long enough to reproduce. I actually was curious why you would think that it would be beneficial, because if an organism lives for a much longer period of time, then they can produce more offspring during that longer time, and then their offspring would have the genes for a longer lifespan and there would be more of them. So why doesn't that result in us living longer?
So it really comes down to what I was saying earlier about the rate of accumulation of damage. That the types of damage that metabolism generates rapidly need to be eliminated, because if you didn't eliminate them, then they would get to a fatal level of abundance before you had reproduced. But if the type of damage accumulates slowly, that doesn't happen. It doesn't exceed the amount that the body is set up to tolerate until we've already passed our genes on. So the thing is that evolution certainly could be better than it is at repairing damage. It could evolve mechanisms and new genes and new gene expression patterns that would further reduce the rate at which damage accumulates in various different types. But if it did so, then we then have to ask, what would happen to those genes over evolutionary time? And the answer is they would mutate away again into oblivion, because there would be no selection for them. The animals that had the genes would reproduce, and they'd be eaten by lions or whatever. But if an animal mutates, has a mutation in one of these genes, so that the new pathway, this pathway that is, if you like, repairing damage unnecessarily well, is no longer there, then those animals are also going to reproduce and then be eaten by lions, and they're going to have the same ability to pass on their genes. So there won't be any reduction in the proportion of animals that have the mutation. So eventually the mutation will just happen everywhere, or different mutations will happen in different lineages or whatever, and you'll end up without your gene. So this is basically why. The more slowly you want to age, the more machinery you have to make yourself age that slowly, and the more machinery you have, the more susceptible that machinery is to mutating, and therefore you end up only with the machinery you need.
That makes sense. I wonder if the brain poses a particularly difficult problem for these approaches. Are the SENS avenues equally applicable to the brain as they are to the rest of the body? Because of course the brain, we certainly understand less about the brain than other organs, and it's by far the most complex one. Plus, depending on what exactly you're trying to do, the brain-blood barrier could also be pretty problematic.
Well, first of all, there is one very obvious way in which, yes, the brain is the most difficult organ to address, which is that every other organ we have, you can grow a new one in the laboratory and just replace the entire thing in one go, with a transplant. And if you did that with the brain, it would rather defeat the object. So that makes it harder. Then you're also raising one other important point, that certain of the ways in which we might deliver a particular damage repair approach involve injecting something into the bloodstream, for example, and letting it just go to wherever it's needed. And there is this thing, as you say, called the blood-brain barrier, which prevents many things from getting out of the bloodstream into the brain. However, that's not an overwhelming barrier, because there are ways in which one can, essentially trick the brain into taking things up like this. There are various techniques that have been used to get past the blood-brain barrier. And once you've done that, most of the problem does become the same as it is elsewhere in the body. Because after all, the brain is made of the same kind of stuff. It's made of cells and stuff between cells, and the cells are made of the same kind of stuff. So yes, it's the same kinds of damage that accumulate, and broadly speaking, the same kinds of approach that can repair the damage that accumulates.
What do you think about potential societal implications for when lifespans are radically increased?
So I spend most of my life reassuring people that the fears that they have about how aging is some kind of blessing in disguise and that it would be terrible if we didn't have it, reassuring people that that's nonsense. But there is one big thing that does matter, which is the transition into a post-aging world. At the moment, people are terrified of getting their hopes up, and even though people like myself are on TV all the time and, talking about progress, nevertheless, most people continue to be terrified of getting their hopes up, and they, continue to try to cling to these irrational rationalizations for aging. And in particular, they cling to the idea that it's not going to happen anytime soon. So, if we get a breakthrough, a really big breakthrough, and, the world suddenly wakes up to this, then the world's not going to be ready. And it's worse than that, because the breakthrough is not actually going to need to be at the level of people living a lot longer. It's at the level of people expecting that they're probably going to live a lot longer.
Being aware of the progress?
Well, being aware of the progress, that's coming. At the moment, you've got people like myself making predictions about how soon we're going to reach the Methuselahity. Very few of my colleagues do that. But that's because they like to play safe, or at least fairly safe. And, that means it all depends on how much progress is made. If there's enough progress, even in the laboratory, not even in people, but in mice, if there's enough progress so that most of my colleagues start to come out and say, yeah, you know what, we're within striking distance, then people are, influencers around the world, the Oprah and Joe Rogan and, Mr. Beast are going to be saying, they're already demonstrating an interest in this area. Joe Rogan's had me and David Sinclair and other people on the show a few times. These people are going to come out and say, well, fuck, we better get on with this, because every day that goes by, there are 110,000 lives that are being lost. And so, influencers, they're called influencers for a reason. So the public's going to be clamoring for this the following week. And that means governments are going to do something the following week. Not just any old thing, but this will be a scale of endeavour that puts the response to COVID in the shade, right? And it's going to happen very, very suddenly as a result of just one or two little sounding breakthroughs that impress the literally half dozen experts in the world who actually have an influence on influencers. There's really very few of us. It's the same as most fields, of course. There's only a few people at the top, and in particular, there's very few people who are A, at the top, and B, talk to the wider world a lot. And so, and I know these people very well, of course, we're all good friends, so I can be very sure that we're quite close to that. So that's what we should be concerned with. We should be trying to anticipate the anticipation, I like to say, and get ready for the time when there's going to be this ultra-sudden sea change in people's expectations and in people's economic choices and so on as a result.
Yeah, and then hopefully such a change would be sort of self-reinforcing, where then more funding pours in as a result, and that'll help move things along even more quickly.
Well, that's the theory.
How bothersome are regulations, do you think?
Much less bothersome than people think. So, at the moment, it takes decades to get a single drug through the regulatory process, from preclinical results through to approval. And it costs a huge amount of money as well, of course. So people will say, well, this is a case where you're going to have to get dozens of things through the regulatory process, and they have to, you have to do tests to see how they work in when you give them to the same people at the same time. It's going to take centuries. And of course, if we were talking about a world in which the attitude to risk, the attitude to safety and so on were the same as it is today, then that argument would have a lot of weight, a lot of merit. But we're not. We're talking about a world in which the world has woken up, in which the public and governments and so on have woken up to the fact that aging is actually quite a bad thing and we can fix it. And so, again, I make the comparison with COVID. We saw what happened there. The quickest that a vaccine had gone from discovery to approval ever before COVID was 10 or more years. And it was done in less than 1 year. So when there's a will, there's a way, and there will definitely be a will.
Speaking of regulations, I'm sure you've heard of Italia City.
Sure have. I was there a year ago.
Oh, really?
Yeah, so the potential for special economic zones like Prospera to move the needle is quite substantial. At the moment, before we get this sea change in public demand, the process of trying to modernize the regulatory structures in established jurisdictions is a huge frustration because there's so many vested interests in favor of the status quo. Some progress has been made in some places, but it's glacially slow. So there's a very good chance that the success of special economic zones like Prospera will be the thing that does move the needle. Because what it'll do is, if places like that become the global hub of medical tourism, and people are going there and getting treatments they can't get anywhere else, and they are reporting very good benefits and so on, and the processes are being done in a transparent way, with results being publicly revealed and with a lot of follow-up and so on. If all that happens, then there's going to be FOMO. There's going to be a case of the public in established places saying, listen, something's got to be done. And that may very well, very substantially accelerate the modernization of organizations like the FDA, which are, at this point, completely not fit for purpose. They were built for yesterday's medicine, for pharmaceuticals. They were built for, administration of single things one at a time. They really are not built for combination therapies, and they're certainly not built for cell therapies and gene therapies.
Do you have an opinion on people like Brian Johnson or the general trend of people doing things like intermittent fasting and meticulously planning their sleep, diet, exercise, etc.? I'm sure it probably pales in comparison to what the actual biomedical interventions will do, but perhaps it's still helpful to try to maximize the probability that a person will make it to longevity escape velocity.
Yeah, I have an opinion about more or less everything. The whole idea of intermittent fasting and related things like calorie restriction mimetics, rapamycin, things like that. As you say, I'm very certain that they won't radically increase anyone's longevity because it's all about activating pathways that are activated with regular famine, in the wild, and the magnitude of the effect is far less in long-lived species than it is in short-lived ones. But even a little bit, as you say, is better than nothing, especially if it doesn't impact your quality of life too much, which of course is very much a personal thing. So, I'm certainly not against these things. I think, I'm all in favour of them, just as long as people don't get carried away and think they are somehow the fountain of youth. When we come to Brian Johnson, okay, in summary, I think that Brian Johnson is a very definite net positive. He's very controversial, certainly, but the main reason he's controversial is because he goes out and tells it like it is. He tells everybody what he's doing and why he's doing it. And a lot of people are alienated by his reasoning. They don't want to live the way he lives, even if they did have $2 million a year to spend the way he does. So that's a problem, but I think it's outweighed by the fact that he has the courage to lead from the front, which is what I've always done as well. So I feel a bit of common cause with him in that regard. All I would say is, if you have got $2 million a year to spend, spending it all on things that are only going to give you a very small amount of increase in lifespan seems misguided to me. And I believe he should be, well, honestly, he should be writing me big checks all the time, which he isn't. But yeah, in general, a definite net positive.
Nice. And then can you tell us about the X Prize, which I read about recently?
Yeah, sure. So, of course, the X Prize Foundation is an organization that's been around for 30 years now. And its first initiative was an X Prize, a prize for space tourism, which was very successful in kickstarting an entire industry that we now have as a result. Since then, they've run a lot of other prizes. I think it's now a total of 30. And one of them is an X Prize to address aging. Now, Peter Diamandis, the guy who runs the X Prize Foundation, has been talking with me for more than 15 years about this. We've been very close friends for a long time. And, you know, for the longest time, we just couldn't figure out a way to design such a prize that had the right kind of mix of achievability and audacity. Essentially, the science wasn't far enough along. But around 6 or so years ago, that changed, and it helped that somebody came along and wrote a small check, well, it's small, $500,000, to design the prize. This is something that the X Prize Foundation always has as a starting point. If they don't have someone giving them $500,000, nothing happens at all. So with that $500,000, they put together a design process, and I ended up fairly soon essentially heading up the biology side of that. And the result is a prize that has my fingerprints all over it. Yeah, it's called a health span prize, but of course, as you can see from the rules, it's really a rejuvenation prize. So the idea is you have to reduce someone's biological age by at least 10 years, and ideally 20, and you have to do it with treatments that are conducted over a period of at most 1 year. So the way in which it's being measured is by looking at 3 types of core function in the body that go downhill with age: cognitive function, muscle function, and immune function. And the rules are, well, quite voluminous, because it's a complicated thing to measure and to assess and to make sure that nobody can somehow game the rules. So I am still very much a scientific advisor to that prize, but at this point I have been joined in that regard by a lot of extremely distinguished scientists, most of whom are more mainstream than me and less controversial, and generally, you know, I'm in a position to be able to take something of a back seat at this point and let the thing really catch fire among the mainstream.
Well, thanks for all that info and insight. Is there anything else you'd like to share, or where can people go to find out more about your work, or what can they do to help?
Yeah, I think we've covered the main things. But yes, absolutely. Anyone who wants to know more should go to levf.org, which you can, of course, put in the show notes. Our website, Longevity Escape Velocity Foundation. And so the study that we've been doing is, of course, documented there. The study that we want to do next, one of them anyway, is documented there, and we'll be updating that over time. And of course, there's a nice big friendly donate button for anyone who feels inclined.
Wonderful. Yeah, I wish you lots of luck in trying to get us to live as long as possible and be healthier along the way. Thank you very much for your time again. I appreciate it. Very interesting, very exciting what you're doing. So,
it's my pleasure. Thanks for having me, and good luck with the podcast going forward.
Thank you.